Hey, hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really glad you made the time to dial in today. We will be talking about the discovery of CHK-336 with Renata Obala today. Welcome to the show, Renata. We're glad to have you.
Thank you. Thanks very much for the invitation. Really looking forward to this.
So Renata, and we were just talking about this, just recently retired after 27 years as a medicinal chemist in the pharma industry. And before that, she was the VP of Research Operations at Chinook Therapeutics. Renata actually co-founded Chinook. She led the chemistry group there, led the research operations, and was also the site head of the Vancouver site, where she is still located today. In 2023, Chinook was acquired by Novartis, mainly for their kidney assets. And their research group, so Chinook's research group, recently has been spun out to form Borealis Biosciences, and Renata is an advisor to that group. Prior to Chinook, Renata was actually the VP of Medicinal Chemistry at Inception Sciences, where she and her team played a key role in the development of small molecule candidates for the treatment of AMD, including the acquisition of an HTRA1 development candidate by Iveric, as well as in oncology with the development of EP2 and EP4 dual antagonist TPST 1495 by Tempest. Previously, Renata was director of chemistry at Xenon and at Merck, and has expertise across multiple disease areas, including pain, anti-infectives, diabetes, and also osteoporosis. She was a key contributor to the development of the cathepsin K inhibitor, odanasertib, for osteoporosis, and also led the team that discovered SCD inhibitor MK8245. Renata received her PhD under the supervision of Edward Pierce from the University of British Columbia, and also conducted postdoctoral research in the lab of Ian Patterson at the University of Cambridge in the UK. So Renata, really, what an impressive CV. You've seen so many things in medicinal chemistry. We're really glad to have you on the show today. So welcome again.
And thanks again for the invitation. My
Yeah,
pleasure.
absolutely. And yeah, in the good old drug hunter tradition, we like to start this with a few questions to get to know you and warm you up for the presentation. So could you share with us what inspired you to start a career in drug discovery?
Yeah, so back in the early 90s when I was in undergrad and grad school, Big Pharma had these great ambassador programs where the industry alumni from the various universities that they had come from would go back to the university and give case study talks on various programs. And so they met with students, including myself, and really got everyone excited about drug discovery and solving complex problems. And so this led to sort of lots of networking events and really understanding, I'm from Vancouver, and understanding which pharma companies had sites in Canada. At that time, they were all in Montreal. And really, these contacts led me to land my first job, where I spent actually half my career at the Merck Frost site in Montreal. And so that was, a great place to really hone my career and start medicinal chemistry. So I think it all, led back to these great ambassadors that would come and talk about industrial positions, whether it's medicinal chemistry or process chemistry, and all the different aspects of drug discovery.
Amazing. Yeah, I think that's super, super important to have those programs to kind of show people what possibilities there are for
Yes,
different careers. I
exactly.
I really love
Yeah.
that. Really love that. And Renata, you've brought so many molecules to the clinic. I just named a few of them in your introduction. I'm sure you have a favorite molecule. Can you kind of share a little bit more about that with us?
Sure. My favorite molecule is actually the first program I was ever on, the cathepsin K inhibitor program, odanasertib. So that was discovered at Merck in the early 2000s. But I worked on this program for 8 years, which is a long time in pharma for medicinal chemists. But it really allowed me to cut my teeth on drug discovery, and as such, it holds a special place in my heart. We started with a peptidic lead with a 15-minute half-life, and this is the time where really drug metabolism was really key in figuring out aspects and really honing and fine-tuning compounds. And so we ended up with odanacatab, which was a small molecule with a 60-hour half-life and really was enabled once a week dosing, which was really key because some of the leading compounds in osteoporosis were once weekly dosing. And so that was really special. And although it made it to phase 3, sadly it didn't make it to the market. But yeah, no, this one will always hold a special place in my heart.
Yeah, no, that does sound really impressive. I mean, if you can make a molecule dose once per week, a small molecule, I mean, that is very, very impressive.
Yeah, no, it was
So
it was a really great program to work on. Yeah.
sounds great. And what are you excited about in drug discovery today?
Yeah, so today, um, there's a lot of new technology and new platforms being built in the RNAi field, and I think targeted RNA delivery, so specifically targeting a RNA to specific cells. There's a number of new biotechs, including Borealis Biosciences, which is heavily invested in this platform, and I think it's on the cutting edge of really different tissue targeting techniques. And so I'm really looking forward to advances in this field.
Yeah, that sounds amazing. I'm glad that we're going to learn a little bit more about tissue targeting from you today.
Yes,
So
yeah.
that is, yeah, I'm
Yeah.
very, very much looking forward to that. I've been working in CNS drug discovery while I was in industry, so that
Yes.
that was always
Yeah.
super fascinating to me.
Yeah, yeah, exactly. That's another layer of complexity in drug discovery when you're going away from a systemic drug and really trying to target it to a specific tissue.
Absolutely, and I think your team has done an amazing job there with the molecule you're going to present on today. So, Renata, our audience has a lot of people that, are maybe a little bit earlier in their
Mhm.
career. So I wonder if you have some career advice for those people, or, maybe rephrase the question, if you could go back to, your grad school times and give yourself some career
Right.
advice, what would you say to
Yeah.
yourself or to your
Yeah.
children?
Yeah, no, exactly. So yeah, and I also wanted to put out there, I do a lot of mentorship now that I'm retired, to undergrads, graduate students, new sort of freshly minted career folks. And if anybody wants to have another discussion with me, just connect with me on LinkedIn. Let me know that you went to this talk and that you're interested, because I'm happy to go that. But my main piece of advice that I'm giving to people these days, especially sort of millennials and Gen Z, is don't overstress about career decisions. I think it's most important to follow your passion, listen to your gut. Your career is going to have lots of twists and turns. I went from big pharma to biotech to operations, and I think it sounds stressful at the time, and it can be stressful, but ultimately can really lead to exciting opportunities that may initially be outside your comfort zone, but ultimately will lead to a really interesting career. And so in my case, it was moving from being 100% scientist to more of a biotech entrepreneur and helping co-found biotechs. And at the beginning, I must admit, the operations side really scared me. I had no experience in that. I didn't know if I would like it. But ultimately found that it was a really challenging part of the job, a really interesting part of the job, and I really liked it. But if I hadn't sort of taken the leap and just jumped to do something like that, so basically don't stress. There's a lot of anxiety, and for good reasons these days. I mean, it's not like when I started in the 90s where basically jobs were raining from the sky. I'll be honest. It's a little bit different now, but I think just you're gonna twist and turn a few times, but don't try not to stress, and just follow your gut and your passions.
I love that. And thank you so much for that offer too. I think that's
Yeah.
super, super valuable to the audience. Yeah. And I know it's very difficult to give generalized advice. So it's much, much appreciated that you're able to give personalized advice as well. Yeah, lastly, I'd love to know, Renata, what do you like most about Drug Hunter?
Yeah, so I think especially for young chemists and students and folks in biotech that, might not have access to a lot of online licenses, I think the fact that you guys, share these flash talks and where you can learn about different modalities and case studies on YouTube, I think is really valuable. I think that's a great resource. And then in terms of other aspects that, people that have drug hunters, sort of the emphasis on practical insights and case studies from real drug programs is very valuable, and sort of distilling a lot of information into these manageable bites that allow people to then be like, oh yeah, I really want to learn more about this. But sometimes there's just too much to digest in the primary literature. So I think Drug Hunter does a great job of that.
Awesome. Yeah, thank you so much. That's greatly appreciated. Yeah, so before we get started with Renata's talk, I do want to show a brief case study, an example for a case study that maybe Renata just mentioned, and that's probably a molecule that's I guess close to Renata's heart. Atrasentan. So what we have approximately 600 to 700 of these case studies available on the platform today, and they grow at a pace of about 2 case studies per week that we add. And the information that you can find in there, typically we start with an executive summary where you have the target, where it's currently at in clinical development. So here, this compound, of course, is FDA approved just recently, how it was discovered, what the last source about this is, and then who invented, maybe discovered, and commercialized it. We start typically with some highlights. We have an introduction of why this molecule is important. You can see these purple links here. There's about over 100, I would say, links in each article, and you can always go to the primary resources with that. We typically talk about the target itself, maybe compounds that have been tested on this target before, so that shows you why it's so important. We talk about the disease itself. We talk about maybe clinical advancement, regulatory advancement, and then in this specific indication, what is the therapeutic hypothesis for this? How is this compound differentiated maybe from other competitors in the field? And then, of course, a lot of really interesting M&A as well. As we've heard before, we talk about the full discovery story from the initial hit to kind of what was the hit to lead, the lead optimization, ultimately to the clinical candidate nomination, the PK data, efficacy data, clinical data here as well. A brief outlook of like a perspective on what this approval maybe means, and then synthetic highlights as well. The cool thing about these articles is we can actually update them when new information becomes available. So we spotted atrasentan actually really early on, and then once there was phase 3 data, we actually updated the article with that information. And then, of course, recently the compound was approved by the FDA, so we updated the article again with more information that became available. So just encouraging people to check out those case studies. As Renata mentioned, they're really kind of valuable to learn more about drug discovery in general. And with that, I'll hand over the stage to you, Renata, and very excited to learn more about the discovery of CHK-336 for PhD, right?
For primary hyperoxaluria. Okay, so before I get into the story of CHK-336, I just want to give a few slides highlighting how this company was formed, why we went after kidney disease, and its evolution. So at the time, the Inception Sciences Vancouver site scientists were tasked with, coming up with an idea for a new company. And, we landed on kidney disease for a number of really important reasons. First of all, it's a very unmet medical need. It's an underserved market. There's very few drugs that have been approved in the past 20 years. And unfortunately, for patients with kidney disease, often there's only dialysis or kidney transplant. Around the time that we were starting, there was some novel genetically defined targets, as well as translational platforms such as single-cell sequencing, kidney organoids, that would help hopefully be able to better or more predictably translate preclinically to clinically. Sort of linked with that was some emerging patient stratification approaches using biomarkers, causal mutations that would help predict which patients in a particular indication would be best suited for a drug. And last but definitely not least, the FDA was then starting to allow some surrogate endpoints as registrable endpoints, especially for diseases where there was a good understanding of the pathophysiology. So for proteinuric diseases that had high protein in the urine that correlated with end-stage renal disease, such as IgA nephropathy, the drug that atrasentan is just recently approved for. They were allowing the use of measuring protein in the urine as a registrable endpoint. Total kidney volume for polycystic kidney disease. So instead of going from thousands of patients in a 5-year outcome trial, you could get approval in 6 to 9 months in hundreds of patients. And for a small biotech, this was really, really important. And so we started in January 1st, 2019. There was 20 people at the Vancouver site. We only had preclinical assets, small footprint, 12,000 square feet of lab and office space. We then hired our CEO, which was Eric Dobmeier, the former COO of Seattle Genetics, and really helped shape the strategy for this company and lead us in fundraising. So he, along with Versant Ventures, our key backers, led the Series A funding in August of that year. Now, we only had these preclinical assets, one of which I'll talk about today, and we knew that these would take several years before they got to IND stage. So we were always looking to see, could we in-license something for kidney disease? And atrasentan was being developed by AbbVie for chronic kidney disease. It had been in 5,000 patients, had a great safety profile, and we really had the idea that it would be really well suited for IgA nephropathy based on some protein urea data that had been reported in a subset of patients. And so we licensed in this asset a year after we were founded, but it was ready for phase 3. So think about it. We're a super small preclinical team. We have to start a phase 3 trial. Look at the date, just before the pandemic. And so we often, as Eric said, we were basically flying the plane as we were building it. So we had to build a huge development team to carry out these trials. We needed more money, so we raised another round of financing. And then, we have a phase 3 asset and we have early assets. And we needed more money to continue the development of an expensive phase 3 trial, but we wanted something also in between. So the development team would have something to work on as this compound was going through phase 3. And so we did a reverse merger with Aduro Biotech in the Bay Area. They had 200 million in cash. It launched us onto the public market. And importantly, it had a kidney asset. It had a monoclonal antibody that they were studying for multiple myeloma. This was an immuno-oncology company, and we thought would be very well suited for IgA nephropathy. And so again, this asset bridged the pre-IND and the phase 3 assets. And so sort of at the end or 6 months later, we'd grown to 110 people, and we had now assets in phase 1, 2, and 3, and preclinical. We expanded the Vancouver footprint. We built a vivarium, we built a DMPK suite so that we could really do all of our research, most of our research in-house. And then we added offices in Seattle and Oakland in the Bay Area where Aduro was from. Okay, so about 6 months later, we formed a joint venture in China because IgA nephropathy is a rare disease, but it has high prevalence in Asia. So we thought it would be important to form a venture there to develop kidney diseases in China. And then about a year later, we started getting some readouts from the clinical trials with both the small molecule atrasentan and the biologic 1301, which demonstrated a very robust reduction in proteinuria, the registrable endpoint for these kidney patients. And then at the end of that year, we had a very strong balance sheet, well poised to continue phase 3 development and start phase 3 for this asset. And we were ready to launch commercialization. We had hired a chief commercial officer, etc. 6 months later, we had phase 1 data for the compound that I'm going to talk about today. This is our first homegrown compound. And we were a small molecule group, so we wanted to forge partnerships with people that were experts in biologics. So we forged a partnership with Ionis so we could go after kidney diseases that would be better suited to ASOs. And at this point, we had grown to 260 folks. And so the phase 3 readout for atrasentan was going to read out at the end of 2023. Novartis ended up taking a bet on us and acquired us prior to the phase 3 readout in August of 2023. So over the next year, we integrated in with Novartis and we transferred all of the clinical and preclinical programs. We filed the NDA for atrasentan, and then very gratifyingly a couple of months ago, especially in this climate with the FDA, we were able to obtain, or Novartis obtain approval for atrasentan. So everybody was very excited about this. At Chinook, I'm sure that folks at AbbVie that originally developed this great small molecule and Novartis. Okay, so with that, actually, before I go, a lot of people ask me what happened to the team. So the preclinical team, as Dennis mentioned, spun out to form another kidney company called Borealis. They are doing RNA medicines. And a lot of the development team are at Jade Biosciences, which is an autoimmune disease, and also studying kidney disease. So great development for all of these fantastic scientists. Okay, so I'm gonna just dive in now with CHK-336 and talk about how we developed this liver-targeted small molecule inhibitor for against LDHA for the treatment of primary hyperoxaluria. So I'll first start with what is this disease? It is a disease that is caused by excess oxalate that is formed in the liver as a result of genetic mutations. So this oxalate is overproduced. It gets excreted by the kidney. It's a small polar molecule, but because it's in such high concentration, it crystallizes out in calcium oxalate kidney stones and basically completely damages the kidney. It's a very serious disease. It starts in children. They develop kidney failure in their mid-20s, and it also this oxalate also spills out to other organs. It's ultra rare, so 5,000 to 7,000 patients, and up until recently, the only way to treat these children, it was a dual liver kidney transplant. So liver because that's where the defect is, and kidney because they've been damaged. So you can imagine having a double transplant is very, very, very challenging. So our goal was to go after these primary hyperoxalurias as our first proof of concept and get into a market for this, and then go after a more common disease called idiopathic hyperoxaluria, which results in patients that have some metabolic diseases that also result in overproduction of oxalate. And this can be found in some of the frequent kidney stone formers. So Just a little bit more about the genetic diseases. So there are three types of pH, 1, 2, and 3. They have mutations in various enzymes, and they result in a loss of function of these enzymes, which all lead to an increase in the glyoxalate to oxalate formation. So in the liver, the last step in the synthesis is carried out by LDHA, and you get this overproduction of oxalate. Recently, there has been two siRNAs approved for the PH1 form of the disease. One is a lumasiran, which targets the enzyme glycolate oxidase, and one, nedosiran, which was developed by Dicerna and now owned by Novo Nordisk, targets LDHA. So we went into this program thinking about making a small molecule inhibitor that could be given orally once a day to inhibit the final and committed step in the synthesis of oxalate, with the hopes that we could treat all forms of the disease because it was downstream of these mutations. Okay, so I'm going to switch gears a little bit to oncology because LDH inhibitors have been implicated in oncology because cancer cells have a high propensity to uptake glucose and convert it and use it as an energy source and converting it to lactate. It's known as the Warburg effect or aerobic glycolysis. So it's not surprising that a lot of pharma companies and biotechs have developed LDH inhibitors because LDH is a key enzyme in this role. It converts pyruvate to lactate. And so inhibiting LDH should reduce glucose metabolism and reduce tumor cell proliferation. Unfortunately, none of these compounds advanced to clinic. There was very limited preclinical efficacy. And, there is the thought that, cancer cells can, if one metabolic pathway is blocked, they can shunt and use another metabolic pathway. So unfortunately, it didn't pan out for oncology. These were, of course, developed for systemic use to penetrate tumors. And so we thought, okay, there's a rich literature of LDH inhibitors. Could we potentially identify a series that we could use for the inhibition of LDH and particularly for the glyoxalate to oxalate pathway in the liver? And we knew two things when we went into this program. One is that there are humans with loss of function mutations of LDHA that have exercise-induced muscle adverse events. So indicating that we wouldn't want a systemic inhibitor because there would be adverse effects due to inhibition of LDHA in the muscle. So we would need to be liver targeted. We knew that going in. And we also knew that Dicerna had just generated some proof of concept data in phase 1 with a GalNAc liver-targeted siRNA, showing that yes, if you knock down LDHA in the liver, you do indeed reduce oxalate, and it was safe and well tolerated. So there was this clinical proof of concept, which is huge for a small biotech, and we really wanted to sort of approach this enzymatically, and could we inhibit this with a small molecule? So these were some of the inhibitors that were in the literature. There's many, many more, but Genentech, GSK, and the NCI Next academic consortium had published, a lot of literature and patent applications in this field. As I mentioned, these were all, systemic, and we wanted to determine, could we convert some of these to a liver targeted so that we could inhibit the target in the organ that we needed to inhibit it in, but minimize any off-target inhibition in other tissues, such as the muscle. So before I start talking about the profile of these compounds, I just want to quickly go over some of the key principles in how to target a small molecule to the liver. And one of the key ways to do this is to engage active transport into the liver utilizing liver-specific organic anion transporters. And this has been successfully employed by statins, although they didn't know it at the time, to inhibit cholesterol synthesis in the liver while avoiding inhibition in tissues such as the muscle, which gave, muscle pain if there was a systemic statin. And so, an acidic moiety is kind of a key. You can see here there is an inverse correlation between the uptake by OATPs and the pKa of the molecule. So neutral or weakly acidic compounds are not substrates compared to compounds that have acidic moieties, such as a carboxylic acid. And these two compounds already had an acid already in the molecule. The other thing that you need to dial in is low passive permeability, because whatever remains in circulation, you don't want it to be super permeable that it penetrates other tissues. And so the sweet spot is generally in an MDCK or a Keiko2 cell assay, 1 to 5 times 10 to the minus 6. You don't want it too low because then it won't get absorbed. So because we were going after an oral compound. Okay, so here's these three compounds that we initially studied. The GSK compound was pretty big, and a little bit not drug-like, reasonable potency. We knew, though, from the SAR that had been reported by GSK that you couldn't really lop off parts of the molecule. You would lose a lot of potency. So we really didn't invest too much time in this high molecular weight compound. The Genentech compound was really nice and small, 500 molecular weight, reasonable potency. It had the right Keiko2 permeability in that 1 to 5 range. Unusually, though, and surprisingly, it had high bioavailability. And what we did was we carried out a tissue distribution study in mice. This became the workhorse for the program, where we would dose compounds and then look at the mouse, liver, plasma, and muscle concentrations. And we went in at a high dose, 400 mpk, because that was actually the efficacious dose in oncology studies with this compound. Okay. And you can see that the liver levels are huge, a millimolar, but unfortunately, the plasma levels were high and the muscle levels were really high. So it didn't really have a liver targeted sort of inclination. The NCI next compound, 63, intermediate molecular weight, exquisite potency. So we were really excited by this. As a result, it had a high LLE, had low permeability, maybe too low, zero 0.0. But, it did have a little bit of bioavailability, enough for us to do some tissue distribution studies to find that it actually had a really nice starting point. There was 4 micromolar in the liver after 1 hour, a 20 mg per kg dose with low plasma and muscle. What the one big drawback is that in a primary hepatocyte assay, where we're monitoring LDHA inhibitor, it's very shifted, 5,000-fold. This could be due to the fact that it had low permeability, as we knew, or the fact that maybe it wasn't a great substrate for the OATPs that are present in these hepatocytes, and there wasn't transport in. So before I talk about a little bit about the SAR, these are the 3 things that we were going after. We wanted to, have this active transport and improve this so that we could have increased concentrations into the hepatocyte, where the LDH was present. A lot of people ask, why didn't we use OATP assays as our primary screen? These transporter assays are actually pretty laborious. They require really specialized conditions. You often need radio-labeled compound, so they weren't high throughput. So we used a primary hepatocyte assay where these OATPs are active, and monitoring LDH as a surrogate for things that would get in via OATPs. We wanted to limit passive permeability, and we could clearly monitor this quickly and in high throughput using Keiko2 or MDCK assays, and we could fine-tune phys-chem properties to help address this. And then finally, compounds are getting sucked up into these hepatocytes. We wanted to minimize metabolism, we wanted to minimize efflux, and we really wanted to have a once-a-day compound. And so we thought, could we bind it tightly to the enzyme of interest, because there was high LDHA concentrations in the hepatocytes, and enable this liver-restricted target-mediated drug disposition. And I'll talk about the primary assay that we use to sort of gauge slow-off kinetics. Okay, so starting with that NCI next compound 63, otherwise known as CHK-569, the compound had very good potency, as I mentioned before. This was just in our own in-house assay under slightly different conditions. And we set about monitoring a series of alkyne inhibitors, not because we like alkynes, obviously they're not drug-like, but they were simply a handle to add various heterocycles and cycloalkyl groups. We knew that this part of the molecule from crystallography studies was relatively open, and we could potentially modulate properties here. We knew we couldn't touch the acid for OATP reasons, but also because it played an important interaction with the enzyme, and this sulfonamide was also very important. So the first thing I want to point out is that, yes, we maintained potency. That was very gratifying. We didn't really change the phys-chem properties in this series of compounds. And the mouse hepatocyte potency, got a little bit better, not dramatically though. But what we did do from this workhorse tissue distribution study was really being able to assay what the liver targeting was. And this compound really stood out. It had high liver levels and very prolonged liver levels. So even after 24 hours of dosing, there was still 5 micromolar in the liver. So we had an idea that these alkyne series potentially had slow off kinetics, but there was very subtle SAR. You could add a heteroatom or two and you lost the liver levels. So this wasn't, you couldn't predict this. You really had to carry out these tissue distribution studies. So in vivo played a key role in doing this. And then, I'm going to summarize 1,000 compounds worth of SAR into one slide, as we do in MedChem. And here we really wanted to decrease the lipophilicity. So we modulated this with heterocycles, heteroatoms, etc., lopping off this group altogether. And you can see potency was nicely maintained, as was in the mouse, which was our efficacy species. And we could get down to about 180 nanomolar in the hepatocyte assay with excellent liver targeting. And this actually became our compound, the simplest compound without anything here, CHK-336. And so we wanted to figure out if this had a slow off kinetics. And so this was the assay that we used to rank order compounds. And it was a really simple assay, but was really great for the program. So the primary assay is, as usual, enzyme, inhibitor, substrate, NADH, you get an IC50 curve. We did a desalting size exclusion spin plate that basically compounds that were tightly bound to the enzyme flowed through. We would repeat the assay without adding any inhibitor. So if the compound was tightly bound, it flowed through, it would have an IC52 that was equivalent to IC51. And things that had a fast off would remain here, and then you would get a shifted IC50. So we ran this alongside the primary assay, and we could rank order compounds. So is CHK-336 a tight binder? So the first panel I want to draw your attention to is just with the wild type enzyme, exquisite potency. We do this spin column, exactly the same IC50, indicating it's slow off and really tightly bound. When we did the crystal structure, we saw that on top of the inhibitor, there was this salt bridge that was formed. We did a double mutant of LDHA, which disrupted this salt bridge. I don't show you the crystal structure, but we've changed aspartic to serine and tyrosine to phenylalanine. And when we run the assay with the mutant enzyme, 336 inhibits it exactly the same as with the wild type. So the key interactions are there and important. However, when we do the spin column without this salt bridge, the compound rapidly dissociates in the IC50. So this salt bridge for some compounds and for 336 basically traps the compound in and results in this slow off rate. So now, we radio labeled the compound. We were able to do the key studies. It was a human OATP substrate shown here. So you have this rapid active transport relative to mock transfected cells in all of the liver specific OATPs. And if you inhibit the OATPs with rifampicin, you decrease this. So this proved in vitro that it was a substrate. In vivo, we did a study. So, the wild type mice have this 100-to-1 liver to plasma ratio, which is great. If we knock out the OATPs, we erode the liver targeting. So the plasma levels are much higher because it's not getting as sucked up, and you erode this. We put in humanized OATPs, and you can again restore this liver targeting. So showing in vivo that we can that the OATP is responsible for the liver targeting. So is the target-mediated drug disposition responsible for the slow off and the long liver half-life? So in gray is sort of the 4-hour and the 24-hour time points in the liver of 336. If we knock down LDHA with an siRNA prior to this experiment, so 3 weeks prior to the experiment, we treat the mice with the siRNA, we knock down LDHA to greater than 90%. We redo the experiment. And when at 4 hours, we have the same liver levels because, of course, OATPs are there. They're sucking it up. But the 24-hour levels are much lower because there's no LDHA to keep the compound in the liver. So the target-mediated drug disposition is indeed responsible for the long liver half-life. So, this is just a summary showing that, yes, the compound gets sucked up into the liver. Because there's high concentrations in the liver of LDHA, it is retained there due to this salt bridge and due to the slow off. What little circulates has low penetration and has very little inhibition in off-target tissues. It's always important when you're doing a tissue targeting to not just rely on mice. We did a number of species, including non-human primates, to verify that indeed liver targeting is present in higher species, and the long liver half-life is also present in. So we were confident going in that we would have liver targeting in humans. These are the preclinical efficacy studies that we carried out. So for PH1, you can knock out or knock down the same enzyme as in humans, AGXT. This results in an increase in urinary oxalate excreted in the urine, just like as seen in humans. And then you can normalize this with once oral daily dosing over a 1 week. So relatively short period of time, you can knock this down. And PH2, there's also a model knocking out this enzyme, and you can also show this. So this, having that important preclinical proof of concept. Again, going into a tissue-targeted compound in clinic where the liver levels are going to be really, the plasma levels are going to be really low, you want to show target engagement to really help define your dose that you're going to go into clinic. And so we couldn't, unfortunately, use just urinary oxalate in healthy volunteers because their oxalate levels are super low. So we made an isotope labeled to study this. So using C13 glycolate, which gets converted to glyoxylate and then by LDHA to oxalate. And then you can measure this in the urine by GCMS. So this was going to be our way of measuring proof of mechanism and target engagement. We carried this out first in rats, dose the compound, dose the tracer, collect urine, and you can see a nice dose-dependent decrease in the synthesis of oxalate as measured in the urine. You can measure the liver levels, you can generate this, and you can measure a liver IC90 of 3 micromolar. And then based on plasma to liver, free fraction in liver, free fraction in plasma, you can predict a human dose of 60 to 125 mg daily. So we went into clinic, and this is the phase 1 studies where the primary objective, of course, is always safety, tolerability, PK. You can show this nice dose-dependent increase with a long half-life. We went up to 500 mg in the SAD and up to 60 mg in the multiple ascending dose for 14 days. No dose-related trends in AEs. However, we did have an allergic reaction in one MAD subject after they received the first dose of 125 mg. But fortunately, it was rapidly resolved after antihistamine treatment. The FDA was fine with us continuing the study. However, we did put a voluntary pause on this for a number of reasons. One, we were gearing up all the phase 3 and phase 2 studies, so our clinical team was super busy. But this was around May 2023 when we were just being acquired by Novartis. But we were able to achieve or to show proof of mechanism in patients. So similar to what I've showed you with rats, we measured the oxalate following tracer dosing in the urine, where we have this dose dependent decrease in oxalate that levels out and basically has maximal inhibition at the 60 to 125 mg dose with an unbound IC90 of a nanomolar, so very low plasma levels. But we were knowing that this inhibition is occurring in the liver. So really big kudos to our DMPK group that predicted this 60 to 125 mgs dose as our predicted dose. So in the spirit of time, I really want to allow time for questions. This is just a quick summary. But really, acknowledging the preclinical and clinical team at Chinook. But I also want to spend just a minute on our academic collaborators who were fantastic. So when we started Chinook, we didn't have any expertise in kidney disease, didn't have any expertise in this rare disease and oxalate metabolism. John Knight at the University of Alabama was instrumental in helping us, with these animal models, helping us understand oxalate metabolism. Todd Lowther at Wake Forest for all of his crystallography work, mutagenesis, and really being a key liaison with the patient advocacy groups as well. And last but finally not least, the NCI Next Consortium that developed the original umbrella series of these great LDHA inhibitors, and we had a really great close collaboration with them, learning everything about this inhibitor class and LDHA in general. And with that, I'd again like to thank Drug Hunter and thank everyone for their kind attention.
Awesome. Yeah, Renata, thank you so much for really an amazing presentation. I mean, this is such an impressive program, in my opinion. I mean, first, with this target that you that came from cancer, that you related to this ultra rare disease, and then the choice of the compound and how you optimized it in such a complex environment where you have to have liver targeting, where you have to potentially measure for almost all compounds, what's the exposure in the liver, which is really complicated. And then, being able to nominate this great compound and showing the clinical data, really, really super impressive, Renata. So want to congratulate you on this fantastic program.
Thanks. No, it was a super fun program to work on, and like I said, the academic collaborations were really special, and we had, prior to COVID, at least we had a lot of face-to-face interaction and just constant communication and just really having everybody at the table sharing ideas and working together to try to advance things for this very very important rare disease.
Yeah, cool. Well, so let's get into the audience questions. Looks like we have some already. So, Yupon is wondering, it looks like you used total liver to plasma ratio as one of the criteria to prioritize compounds. Have you collected any KPUU data? And do you think that may guide you to a different candidate?
Yeah, no, we were measuring, whole, but we were also measuring unbound plasma and liver and KPUU. So I think I didn't show a lot of that data, but yeah, we definitely utilize that so I don't know if we would have come up with, I mean, I think we were already using that. So to answer the question, yeah, it was a big drug DMPK lift for this program. Big kudos to them. This is a complex issues, but liver targeting, it's not like it was back in the 90s or early 2000s. There's been a lot of great advances and I think now moving to other tissues is the next challenge, as well as with RNAi. But yeah, it's a fun program.
Yeah, no, totally. And the other question that I was wondering about, you showed that translation later. I mean, of course, in kind of the lead optimization phase, you were looking into basically liver, muscle, and systemic exposure in mice.
Yes.
But of course, you ultimately showed that, and that it's also, liver targeting in different species. In general, from your experience, do you think mouse is a good proxy for, people working in the, maybe liver targeting community? Is mouse a good proxy to look into
Yeah.
targeting?
I think I've worked on two liver targeting programs in my career, and we did use mice. I think it depends on the target, right? So we were also looking at target-mediated drug disposition. So how much concentration of LDHA there in the mouse liver relative to rat, relative to human? Fortunately, it was similar. So we had that. So I think it all depends on, are there species differences? I think it's always important, to start off. We were doing mice because that was our efficacy model, and obviously it also uses less compound. We were having to do all these in vivo studies, but it's
Mm,
important to check early on in rat, make sure there's no discrepancies, because rat also is going to be your safety species. And then as early as you can, run a higher species, right?
yeah.
Just especially for the whole OATP field. And you could do a biopsy study in monkeys, so it doesn't have to be terminal, which will be less expensive. But in my experience, the mouse was pretty predictive. But I think early on, you want to test a key compound in a couple of species, at least in rat, and make sure that that's okay. Yeah.
Yeah, that's I think that's great advice. And Renata, with the respect to the expression of those transporters
Hmm.
in different species,
Yeah.
is there, large differences, or would you say the expression levels are, similar in rodents to non-rodent species?
That's a good question. I don't know about the levels. I mean, the obviously the identity of the OATPs is slightly different. But fortunately, you can buy these humanized OATP mice. So that is something you could quickly check. You could do
Yeah.
the wild type mice versus the OATP and see if there's any difference. But in terms of expression level, you know, I don't know actually. I
Yeah.
imagine it's sort of similar because we obtained really similar results with this compound, but I think it also depends on more, if you're a substrate, how promiscuous of a substrate are you in that you would inhibit both or be a substrate for both mouse and human. But it's something you can easily check out, fortunately, due to the availability of these mice.
Yeah, makes total sense.
Yeah.
Well, let's switch topics a little bit. So Sam from Ventus is saying, "Hey Renata, great talk, thank you. I was wondering on what basis Chinook was able to be confident that inhibiting LDHA only in the liver would preclude the muscle effect seen in people with LDH LOF." Maybe
Right,
we'll
with the
start
loss
there.
of function. Yeah, no, it's a good question, because the obviously the siRNA that was developed was exquisitely liver targeted, right? So it was safe, but we are still obtaining some AAs. So we basically did a lot of safety studies early on, and did some sort of non-GLP studies and push the dose so that we would have a big margin. So I think we didn't know exactly how low of a level we needed in the muscle, but by carrying out these preclinical safety species, making sure that we didn't have muscle or other adverse events early on in the program was sufficient. We didn't know how low. sometimes you get pulled into the ones that have no systemic exposure, that they only go to the liver and it's extremely low. But those compounds are hard to develop because you can't monitor, you can't look for dose proportionality. It actually makes, if it's too targeted, it makes development difficult. So yeah, it's a sweet spot.
Yeah, totally. I was wondering, you alluded to it a little bit, the kind of dose projection for a
Yeah.
liver-targeted compound. I think that seems very complicated. Could you elaborate a little bit more on that? Do you base that typically on liver exposures rather than systemic exposures?
Yeah. So yeah, preclinically you base it all on liver exposure. But then you sort of back calculate to say, okay, this is the plasma levels that give this liver exposure. And then you do a bunch of preclinical species to look at the liver to plasma ratio. So we had that for monkey. So then you predict, what's the ratio going to be in humans, and then look at free fraction in human plasma versus rodent plasma, and also free fraction in liver homogenates, and then do this, back of the envelope, not back of the envelope. It's actually a very complex calculation. I have a slide on that, and I didn't include it because personally, I need my drug metabolism colleagues to walk us through. But I think the key though, you're always going to be a little bit blind going with the plasma level, right? Is it really going to be 400 to 1 in the liver? And that's why this target engagement is so important, right? Because that target engagement is you give the tracer orally, it goes into the liver and it gets converted in the liver by the enzyme, and then the product gets excreted. So by measuring that and looking at what's the dose that gives the highest amount of inhibition, that's actually what you ultimately base your dose prediction on, and guides you in the dose selection. So it's super important to have this target engagement marker for tissue targeted compounds, like PET scanning for brain targeting, same kind of thing.
Yeah,
Not PET, PET tracers.
that
Yeah,
traces. Yeah, yeah, yeah.
yeah.
Yeah, super cool. Stefan is wondering, I assume the acid is getting glucuronidated. So is there any like in terms of the kinetics, or did you actually see
Yeah,
glucuronidation of
yeah,
the
we
acid?
definitely, as you would expect, we
Yeah.
definitely do see glucuronidation. And we did measure, obviously in safety species, we didn't see it being saturated. Like it gets converted in the liver and then it gets excreted. You do have a little bit circulating, and I don't think we did see saturation, if I remember correctly. Yeah.
A saturation of the OATPs, you mean?
Yeah, yeah,
Okay. Yeah.
yeah,
So-
yeah,
Yeah, cool. That was the second question. And
yeah,
then Zaki Cruz is wondering, are there any issues with patentability with this compound with respect
yeah.
to like the NCI
Yeah.
compounds?
So, okay. So yeah, that's a good question. So when we went into this program, we were looking at all these literature leads, and the NCI Next patent family is a great, rich patent family. I think there was 1,000 compounds. And so we could have taken a tact of like, okay, we're going to go outside the IP of this great series, but that adds another hurdle, right? Where you're really having to carve out space. So we licensed in the general umbrella and worked with the NCI Next, licensed in that application. And 336 itself was not ever made in this umbrella. It was we designed it for the liver targeting, but of course it's generically covered by this patent umbrella. So we licensed in the patent, and then we filed a specific patent application on 336 and other compounds that were not originally claimed. So there is composition of matter patent with this, but of course you have to have the umbrella patent. So that was such a great way to collaborate and save us, the effort of having to patent bust and go outside of this. And the NCI Next really, they had been working on this for a long time. They really wanted to see their series of compounds go somewhere. So I think it's a good example of how biotech and academia can work together.
Yeah, yeah, really, really nice. Yeah, Renata, maybe one last question. I found it fascinating how you were able to design such a nice compound with the slow off rate. In general, do you have any recommendations for people that are working on targets that require a slow off rate? How, like, I mean, the assays that you showed are amazing, I think. Any like tips for
Yeah.
designers of how to design in a slow off rate?
Yeah, I think if you start dialing in, picomolar potency, slow on, slow off, you generally kind of like if you get into that really potent range, you generally sort of land into that. But doing kinetic studies to measure off rate, again, very challenging. So that's why this simple enzyme assay with the spin column was a way for us to rank order compounds, compounds that had the equivalent IC50 versus ones that were tenfold versus ones that were 100-fold shifted. So that just gave us a really quick insight and okay, what are the structural features that give us this SAR? So that's what that practical assay enabled us.
Yeah, sounds good. Like, yeah, maybe we have time for one more question about hepatic recirculations. Did you see that as a
Yeah, we saw it a tiny bit, but it definitely wasn't in this case was not. It can be. It can give you that extra hump and give you a longer drug exposure. In our case, it we really believe it's due to limited metabolism and this tight binding to LDHA that really provides the long liver half-life.
Yeah, makes sense. Well, Renata, thank you again for an amazing presentation and like sticking around to answer all these audience questions. Really, really appreciate it. So
My
yeah,
pleasure. Great questions. And again, if anybody wants to reach out for career advice, happy to give it.
absolutely. Thank you so much. That's greatly appreciated as well. And yeah, we will be here back not next week because next week is actually a holiday, but in 2 weeks. And we will have Phil Barron here talking about simplifying synthesis with radical cross coupling. So that'll be an exciting topic, especially for the synthetic chemists among you. And with that, I wish everybody a wonderful day. And thanks again, Renata, for an amazing talk and a great Q&A.
Great. Thanks so